Hello, and welcome to today's webinar, Current State of COVID-19, Point of Care Testing, Blood Gases, Biomarkers, and Future Directions. I'm Kristen Durr. I work at the American Association for Clinical Chemistry, and I'm here just to share some simple housekeeping reminders before we get started. Before we begin, I would like to remind you that this program is accredited for Accent Continuing Education credit. At the conclusion of the webinar, you will be directed to a link to complete a short evaluation, and you will be able to claim your credit then. Please note that we will conclude with a question and answer session after the presentation. Feel free to submit your questions at any time during the webinar through the Q&A dialog box on your screen. It's in the bottom right-hand corner. However, questions may not be answered until we have reached the question and answer portion at the end of the program. So let's begin. We are delighted to bring you this educational activity presented by AACC and generously supported by Radiometer Medical APS. Today's webinar presentation, Current State of COVID-19, Point of Care Testing, Blood Gases, Biomarkers, and Future Directions. I'm happy to introduce today's moderator, Dr. Larissa May. Dr. Larissa May is a professor of emergency medicine and director of the emergency department and outpatient antibiotic stewardship at the University of California, Davis, and is a national expert in antibiotic stewardship in the emergency department. Dr. May received her MD, her MSPH in Public Health Microbiology and Emerging Infectious Diseases, and her MSHS in Clinical and Translational Research from the George Washington University. She completed her residency training at the George Washington University in 2006, where she served as chief resident. Dr. May's research interests center on the application of rapid molecular diagnostic assays, behavioral economics, and clinical guidelines to improve antibiotic stewardship and other patient safety and quality improvement efforts in the emergency department and urgent care setting. Dr. May has served as an investigator on multiple federally funded and industry-funded trials evaluating antimicrobial stewardship strategies in the emergency department and has published over 60 peer-reviewed articles in her field. She has also served on numerous committees and task forces served in antibiotic stewardship, emergency preparedness, and infectious diseases surveillance for the Centers of Disease Control and Prevention, the National Institutes of Health, and professional organizations, including the Infectious Diseases Society of America and the Society for Healthcare Epidemiology of America. Dr. May?
Thank you for the introduction. I'm delighted to be here. I'd like to introduce our presenter today, Dr. Tran, who is Professor and Senior Director of Clinical Pathology at UC Davis Health and one of my colleagues. He is also the Director of Clinical Chemistry, Special Chemistry and Toxicology, and Point of Care Testing. He completed his PhD degree in Comparative Pathology in 2008, followed by postdoctoral training under the National Institute of Imaging and Bioengineering, sponsored UC Davis Point of Care Technology Center, focused on developing novel molecular technologies for bedside infectious diseases testing. These studies translated to a large multi-center trial evaluating the clinical impact of rapid molecular testing in sepsis in severely burned patients, and now real-world applications for COVID-19. As the clinical pathology director overseeing the COVID-19 testing response, he and his team were responsible for deploying no less than 5 SARS-CoV-2 molecular and serology assays over a 60-day period. In addition to his role at UC Davis, he also serves on the California State Governor's COVID-19 testing task force. We are delighted to have you present today.
Great. Good morning, everyone. It's a pleasure to present with you all and other colleagues amongst the AACC. It's kind of a little bit of a bummer this year that, we can't all gather together at AACC, but we'll be looking forward to it later this year. So, as noted by Dr. May, we're really going to focus on COVID-19, but really center in on the topic of point of care testing and as it relates to blood gases and a lot of biomarkers really and kind of where we go. And, this crystal ball, trying to see where we're going is critical to our operational planning and also ensuring the community that we have means to address this pandemic as it evolves through the course of these critical fall and winter months, where we will have a potential increase in testing rates due to respiratory tract infections such as flu and the regular cold and, of course, COVID. So we have a couple of learning objectives that I'll summarize here. The first is we kind of want to talk about COVID-19, how it has impacted acute care settings, as well as the use of point-of-care testing workflows. We want to apply updated knowledge from published evidence regarding point-of-care testing parameters that may predict outcome or help us prognosticate COVID-19 patients. And then we want to incorporate best practice recommendations for the diagnosis and management of patients with COVID-19 from a clinical chemistry perspective. As you all have experienced since February or March, every morning we wake up, the virus teaches us a little bit more. So all of these, I'll try to chime in that, these are where it's at as of today. And every time I do a talk, I do check in the morning to see what has changed, unfortunately. That, as we know, the world of the COVID-19 evolves very quickly. The biggest issue that we, of course, are living with is that this pandemic continues to grow. And as you recall, back in late 2019, we all heard about this novel virus in Asia. It started to spread. There were closures of cities. And of course, you see the virus continue to spread both east and west from there, and of course, making its way into the United States. And unfortunately, in the United States, we do have a very large number of individuals that have COVID-19. And this has, created significant problems, both medically, but also operationally within the healthcare setting. In fact, if you look more closely, I tried to pull this last night, and unfortunately, as I know, the number keeps changing, but as you can see here, the number of cases in the United States is over 6.8 million, and obviously you all who have watched the news, you've seen that we have passed the 200,000 deaths mark recently. So this is not a situation where we see numbers decreasing. We still see numbers increasing, which is not where we want to go. And states have disproportionately encountered more COVID cases than others. And you can see here, us in California, we do have a lot. We do have a lot of population in the state of California. And population densities, infection control measures, and all that all come into play that help dictate which states have greater testing demands and patient care demands related to COVID-19 than not. So what are our testing needs? You've already heard me mention testing demands, strains on our testing system, and so forth. And when we talk about that, we really have to kind of think about what is from a lab perspective, what's the risk of bringing these tests? What's the risk of COVID-19 in the laboratory? So if you recall, for those back in 2014, we dealt with Ebola. What's the risk of bringing a sample into the clinical lab? Should we go point of care? So, as early in the pandemic, a lot of us experienced, unless you were a well-equipped lab, you had to start thinking about, should we buy these expensive systems? They're not cheap. And you can see on the left right there, this wonderful table that summarizes from the IFCC by Dr. Lippi et al. These common laboratory parameters that are abnormal in severe COVID-19, that predict severe COVID-19 at least. You can see a lot of these things we already have, and we'll circle back on these a little bit later. So it did help us early on that we did have options on the table to be able to address COVID-19 and as well as managing these patients. A lot of us in the laboratory setting, we kind of look back to 2014 to take lessons learned and perhaps the resources we've obtained from Ebola. And so I started my a role in clinical chemistry at UC Davis and point of care in 2014 actually, and my first 2 months was actually oddly Ebola. And so we went from a relatively modest point of care testing operation to this, and you can see this figure right here that we quickly adopted a variety of point of care tests. And if you recall, with the Ebola virus, the high viral loads in blood and other biological fluids really required us to deploy at the point of care, because bringing it into our laboratories, bringing it into the automation lines would effectively contaminate the entire system, and we would have to evacuate the lab and the testing for the entire hospital goes down. You can see that we were able to deploy point-of-care coagulation testing, hematology testing. We also took the jump into going with molecular, including with the Ebola virus PCR test. And we actually brought in a blood culture system to our satellite Ebola lab that was very close to where the patients were treated. And we brought in other point-of-care tests, rapid antigen tests in this case, to be able to look for other diseases that may present similarly to Ebola. The challenges with Ebola, of course, is on the far right there. The evolving PPE requirements, and at the picture below, that's me donning the PPE. So I was one of the first folks to test that out and make sure that these point-of-care solutions can still be used while wearing effectively our spacesuits at that time. So very unique times, but of course, COVID-19 and the SARS-CoV-2 virus is a lot different. So often we don't have to wear that much PPE. PPE is still critical to management of these patients, but our operational needs are a lot different for this pandemic. So what was UC Davis's response to COVID-19? And a lot of us all remember February 29th quite well. We probably spent the first parts of January and early February chomping at the bit to determine what can we do and to prepare. And I would say that in November and maybe early December at our institution, we started increasing our supply more than normal of the respiratory panels, as well as our point-of-care molecular flu and RSV kits, knowing that at least at minimum, if this virus starts coming over the oceans to the United States, we would see an increase in testing to at least rule out other pathogens, more common pathogens. However, as numbers increase in the United States, and the CDC kit that was released early February was then withdrawn due to not working. So we had no ability at public health labs in the country to test for COVID-2 quickly. We kind of started shifting focus on how do we get a test deployed? Do we need to deploy a laboratory developed test, or are there other means? And fortunately, the FDA on February 29th so-called relaxed the regulations. It was more so releasing the template and allowing academic hospitals, other clinical labs and commercial labs to be able to submit their tests for emergency use authorization, or EUA. And that's when the clock started. And I'm sure a lot of us went on to various websites to buy instruments that we may not have. And in our institutions, we were very lucky to have quite a few instruments already. In fact, some platforms that were just acquired just the end of the previous calendar year that was in process of being validated for our regular tests. So we immediately shifted these over for COVID-19. And you can see on March 19, we deployed our initial capability of SARS-CoV-2 testing on a CDC-based assay. We had the ability to do about 20 to 40 tests per day, with the ability to scale a little bit up from that as we expanded shifts, but limited by, of course, the ever so limited PCR and extraction kit reagents. We're very lucky that about 9 days later, we deployed a high throughput system using an EUA kit. So that bumped up our capacity significantly. And as we advance forward into April, and this is over about 60 to 90 day period now, is that we deployed serology, one of the first serology tests at our institutions. We had subsequent others. And we brought in a second high throughput system because if we needed to scale, but more importantly, if you have one instrument, that instrument goes down for many reasons, or maintenance, you're not testing. So we knew we had to have a backup up there. So that helped us deploy. And last but certainly not least, we deployed a faster SARS-CoV-2 assay in our core lab, which is handling the ED. This platform we've had actually for a while. It's actually for our respiratory panel, and we actually had started validation in March, but we didn't see the need yet to deploy it until later because ultimately our strategy was to get initial capability, scale, diversify, and speed up. And right now at UC Davis, we're at the speed up phase as we start heading into the fall and targeting specific populations where timing does matter. And now technology has caught up, and we'll talk about that. In the state of California, we see a great deal of ramping up, and this is from our University of California Health data. University of California Health is the entity that oversees the 5 University of California schools that have med schools and hospitals. As the University of California as a whole, we're a 10-campus university system, but the 5 campuses, UCLA, UC Davis, UC San Francisco, and UC San Diego, are pretty much the centers of testing here, and you can see in the bottom left-hand corner, we strategically cover the high population density areas across the state to be able to address testing. And you can see here that the number of patients we see increased very quickly over time, as well as our testing capability. And the UCs, the 5 UCs, were pretty much the first entities in the state, in addition to our other colleagues at Stanford and USC, to be able to deploy testing. So academic hospitals led the way in the state of California to address this pandemic. UC Davis has, unfortunately, or at least fortunately or unfortunately, has the opportunity to had a lot more motivation to deploy quickly. So for those that kept an eye on the news, and it feels like an eternity ago, February 26th, 3 days before the FDA relaxed their regulations, was a very important day for us. And UC Davis was the institution that encountered the first known community-acquired case of COVID-19. So unfortunate that we were the first in that regard, but fortunate in that it really helped not only change our prioritization in an institution just to get things going, because everyone still felt that at the time this is something that was just slowly brewing coming in from across the ocean. But really this helped us get going very quickly, as you can see with our timeline, but also it did refine the CDC guidelines for testing and amongst others out there. So it definitely changed national direction on this as well. This paper was published in Clinical Infectious Diseases by our infectious disease team and pulmonary team. So these are the folks that managed this first patient. So the patient was in their 40s with no travel history to high-risk countries. They presented to an outside facility 3 to 4 days with flu-like symptoms, and of course their chest x-ray showed consolidation and so forth that indicated that this could be something that we need to take a closer look at. And most seriously, they developed ARDS and septic shock, which prompted them to be transferred to us at UC Davis. This is from the paper itself, and you can see that over the course of time, they got worse and worse, and they finally decided to try to transfer them by the end of day 2 to us at UC Davis for consideration for ECMO to manage their ARDS and other organ dysfunctions. Due to their presentation, our team here requested COVID-19 testing. As you all heard, at the time, there was no local COVID-19 testing. This is pre-February 29th. And at the time, the only place in the country testing was the CDC. So we obviously had to ship it over there, and they had to approve it, and they did not. And so and this was based on guidelines at the time, and as you've all experienced in the last many months, the guidelines evolved. And so you can see here that finally, by day 6, the CDC finally approved the test, and unfortunately, we identified that they were truly COVID-19 positive. They were started on remdesivir, which at the time did not have EUA yet, so this was an experimental drug for testing, and fortunately, the patient did recover. So that was a very important event for us because we now knew that we needed testing, and COVID-19 is now here in the United States, or at least in our region, spreading. So quick overview of the virus itself and the structures and how it relates to targets for us in the diagnostic side. This little cartoon I drew up, and the virus is an RNA virus, as you can imagine, and typical has envelope proteins, nucleocapsid proteins, and importantly the spike protein, which facilitates binding to the ACE2 receptor and helps with facilitates viral invasion. The genome is known as an RNA genome. You can see the various genes that could be targets there: open reading frames, spike, envelope, and N genes are also there that can be targeted. And what we target, of course, with RNA is, of course, molecular approaches, and specifically and most commonly reverse transcriptase real-time PCR. There are many, many other methods that are out there now that are still evolving. There are even isothermal approaches. So don't want to bias towards RT-PCR, but RT-PCR is our bread and butter. It's not necessarily new technology, as you all can remember, and but at the same time, it provides very sensitive means to detect viruses and serves as really the gold standard in many ways to detect viruses, as we know with HIV, HCV, Zika, and of course Ebola, and now we see with COVID-2. The challenges with PCR is that look at the different manufacturers and so forth. You can see that there's different targets. So the CDC kit targets the N region, so several targets within this region. You see a different company target open reading frame regions. Different countries target different regions, including the S gene. So what this means, and what for most of us didn't realize, and a lot of people who aren't virologists and microbiologists didn't realize, is that these genes may have subgenomic material that is present in different degrees of concentration as the virus replicates. So this could affect the performance of these assays, and of course makes it a little bit challenging to compare one assay to another. And we'll speak briefly on how the FDA has tried to improve that ability to compare performance. But it's quite fascinating because these viruses will have different ratios of these genes, which will of course affect your assay. And you can see that here with the 4 assays, and we're now moving towards adding our 5th assay very soon at Davis. But you can see our different assays, and we use a preferred type of control for this, so that we can, at least for our institution, to quantify limits of detection, sensitivity and specificity amongst others. And really, when I say sensitivity and specificity, I really mean positive and negative % agreement, because there is no true gold standard to really compare yet. But you can see here our 4 different platforms at UC Davis have different turnaround times. Some platforms require batching. So our high throughput platform needs to batch 94 samples at a time to be efficient, while others are random access and so forth. But you can see that limits of detection do vary, and sometimes they're reported from the IFU in different units. So this makes it a little bit challenging in terms of that. But for the most part, these tests are very specific. False positives can happen. I don't want to rule that out. And their sensitivity is good. But as we headed into the pandemic, there was a lot of confusion in terms of the sensitivity story. And within 2 weeks of us deploying COVID-2 testing at Davis, questions arose quickly. We saw papers in China and so forth that said that these PCR tests have a 30% false negative rate. Why are you saying 90% sensitivity? It has to be 70%. And with that belief, and immediately within a patient showing up, you can see here that we have an individual here, an elderly lady, so they're high risk, admitted for shortness of breath with pre-existing COVID-19 risk factors. They're on the mechanical ventilator, and they had serial PCR testing, and they were negative, then became positive, then became negative, then became positive again. You'll note that day 1, they decided to test the person twice because they didn't believe the result. And immediately there was a lot, this assay, UC Davis's assay is not good. But we had to spend about a month of our time probably to educate folks on sample quality matters and viral load matters. You can have the most perfect PCR test on the planet, and if that can detect a single strand of RNA on that swab, but there's no RNA on that swab, I'm not detecting anything. So when you put it in that context, people feel a little bit better. With that said, I'll fast forward. We've done over 40,000 tests since then, and our clinicians are now very comfortable with our assay, so that's the good ending to our story. But the sample type does matter. We'll start off with that because viral load matters. BAL, of course, has lots of viruses on average, about greater than 10 to the 8 copies per ml. Nasopharyngeal swab is the next best, at least in literature. Oropharyngeal swabs is the next best. And saliva is now growing in popularity. You'll see papers saying it's great. It's better than nasopharyngeal swabs. Some papers say it's worse. There's a great study from a reference lab in Utah that showed that it does compare well. I am a believer that saliva is a decent sample to use, but I think there's a lot of variability to it. And that's why we're seeing a lot of papers saying something either direction. And in this case, the data we've cited here shows variability of viral load from 10 to the 4 to 10 to the 8. So viral load does matter. And of course, not everyone can get a BAL. So at our institution, we've stuck with nasopharyngeal swabs, but we're looking at other alternatives as the pandemic evolves. Viral kinetics matters. So over time, the virus will shed when it sheds because of your physiology or just because of the virus itself. There's a discussion that the viral load peaks within 2 to 3 days before symptoms. So by the time you see the patient, they're on their downward trend. It's possible that you've tried to detect someone too early, right at the time of infection perhaps, and they're asymptomatic, and you won't detect anything. So they could be below the detection limit. And there's discussions that asymptomatic patients have higher viral loads than symptomatic. So there's a lot of still controversy and discussion there. But at the end of the day, PCR is problematic in finding patients. You can see here these cases on the right with these plots, with the y-axis being viral load, x-axis being days. These are from South Korea. You can see that even over the course of days as patients are recovering, the viral load goes up and down. At some point on day 9 on that orange line right there on panel A, you can see that there's at least one occasion where the viral load was not detectable, and in the next day, on the next day, you'll see that, it goes back up again. So the other thing that we're going to have to come to terms with now is co-infections. I will tell you that a lot of institutions early on during the pandemic back in March and April, if you were flu negative, then, you're going to go get COVID-2 testing. If you're COVID-2 positive, we would hope that was the only virus. But of course, biology is much more complicated than that. And co-infections do happen. And of course, they were recorded. But now with respiratory panels now becoming available with COVID-2 and at least duplex assays where you can do COVID-2 and flu A/B coming online, we'll be able to test that in the fall. And we see here that about 7% from a meta-analysis of hospitalized COVID-19 patients had bacterial co-infections. About 3% had viral co-infections, with flu A, RSV being the most common. So these respiratory panels are probably going to become more important during the season, but that does complicate places that are trying to do pooling, where they are combining samples to get more bang for their buck. But this is something we're going to have to address, because pooling relies heavily on prevalence, and of course, as flu prevalence goes up, it'll throw you off. But we're now seeing the emergence of many point-of-care COVID-19 respiratory pathogen testing systems. And you can see here, these are examples. We have a few of these at UC Davis already, and you can see that these could test quite 18 to 21 pathogens perhaps, and there's now point-of-care respiratory panels. One was recently received emergency use authorization just 2 weeks ago, and so these are going to play a huge role, reagent limiting, of course, as we head into the fall. So we spent a portion of that time talking about the front end. So diagnostic tests, some of this can be used for epidemiological surveillance. And so we're now going to shift gears briefly to now talk about the tail end, to talk about what happens when you recover. And of course, this enters the world of immunoassays, and we're going to first target serology, and we'll briefly talk about antigen testing, which is then back to the diagnostic realm again. So back to the viral structure. When we talk about serology testing, we're talking about host response, and specifically our ability to generate antibodies against the virus after infection. So we can see here, this is now you recovering from infection, and the antibodies you produce target various parts of the virus, including the nucleocapsid, which is in higher abundance, which should improve sensitivity of these assays. But also, it's important to look at high-affinity antibodies. These are the antibodies that are produced that are better targeting the virus, that may correlate better with immunity, and then also targeting antibodies targeting the actual spike proteins, and specifically the subdomains of the spike protein that prevent viral invasion. And perhaps those are the neutralizing antibodies that prevent infection and perhaps confer immunity. And again, the jury's still out. We're still learning about immunity as vaccine studies evolve. This is the classic cartoon of what happens when you get an infection, in this case with a virus, and hopefully and antigens show up, viral RNA shows up early. There's a window period where you may not be able to detect any antibodies, and after about 3 to 7 days, I put 7 days on this cartoon, you have your initial production of IgM antibodies, which are early but not specific. And then days later, you'll have the more specific IgG, which is what you would want if you were to try to build immunity towards this. But we'll add more to that in terms of that topic. At UC Davis, we ended up deploying 2 serology platforms. The first one shown in the first row right there, and that was one of the first laboratory-based systems. We were a very early, unfortunately, naysayer of those rapid lateral flow tests. We knew those weren't going to be great, and we definitely didn't jump into that rabbit hole. But we jumped in early, not because we wanted to, that we felt serology was going to be the big winner, but it helps us know how widespread the pandemic was, especially amongst our healthcare workers. And Dr. May, who's our moderator, is actually our PI of that project. But you can see here that our first assay targeted IgM and IgG. It's a good assay. It did its job. But we've since switched over to an IgG-only assay that targets the S1, S2 subdomains of the spike protein. so that we can have a better clarity of potential neutralizing antibody production in this population. And we've been happy with both assays since then. And you can see here, going back to a similar cartoon of when IgG and IgM show up, we don't really know what happens after a month and what it means. And, with our study at UC Davis, again, led by Dr. May, we actually go out several months, and we can see that folks actually have different responses. And these are their responses over a 3-month period. You can see a decreasing trend. And we actually looked at 4 different platforms at UC Davis. And you can see here that the decreasing trends are not surprising, but at the same time, you see different assays have different performances, and one assay in particular actually gets very close to its detection limit. And in fact, there's a good number of patients that were tested with this platform that had undetectable antibodies by month 3, whereas the other assays, especially those that target high affinity antibodies, as well as those that target the S1, S2 domains, they are still detectable, which makes you feel a little bit better, but also suggests that we now should be more interested in different antibodies and so forth. And I do want to throw out there that antibodies are only part of the picture. We may start hearing more about T cells next, and with these vaccine studies that are going. So our recommendation to our institution was, if you're going to try to test symptomatic folks, have folks return to work, test asymptomatic assays for pre-procedure screening, you're doing PCR. It's going to be that MP swab at Davis. And other places may use different types of swab techniques. Asymptomatic patients altogether, and you want to make sure that maybe they've had exposure or not. You can throw in a serology test, but take it with caution, and especially return to work COVID-19 positive patients. Special screening cases, if you're doing community screening, looking at the epidemiology, looking for convalescent plasma donors, false positive workups, which we'll talk about, prior PCR positive workup, and then those children with the multi-inflammatory syndrome cases where they're PCR negative. That is where serology can play a role, and that's effectively, in part, what CDC guidelines recommend. The false positive workup is pretty interesting here. And you can see that we had an interesting case at our institution where a patient was admitted to our COVID-19 unit after testing positive. They had minimum symptoms, so they decided to retest because folks thought it was perhaps a false positive result. And they determined that it looks like it's a false positive result, so they decided to remove them from isolation. 8 days later, we ran SARS-CoV-2 testing, and it was performing. It was positive, and it was positive for our anti-spike IgG. And on the right there, we also then correlated this with a small cluster that was identified, a very small cluster with 4 additional individuals. And we'll zoom in. And this is actually sequencing that was done for these PCR positive results from that employee cluster. You can see that the virus was very similar. We're able to see how many mutations occur, and there's only a few mutations occur after each transmission of the virus. And so if therefore the mutations are very few from the original sentinel case, it's very likely that came from that one population. Now I'll zoom in right there. And this was done by our colleagues at UCSF, the Tran Zuckerberg Biohub. So they've been a wonderful partner for us at UC Davis, but also for the state, because they've been doing this with public health. So it's been very interesting to see that, but also emphasize that 2 things, that our PCR test at our institution has very few false positive cases. I don't recall one anytime recently, but at the same time, serology can also help you determine if it is a false positive or not, because if they didn't have the antibodies, but then they did, chances are that PCR result was real. So the next thing I want to focus on briefly is antigen testing, because that's starting to pop up on our radar. So instead of looking at antibodies that our body produces against the virus, it's now using antibodies that you've engineered, so immunoassays, to target the virus particles, its proteins itself. As of September 2nd, and actually I've looked recently, there's about 4 antigen tests that have received emergency use authorization, and predominantly they are EUA approved for testing in symptomatic individuals within a certain amount of days of symptom onset. So they have not received that FDA label to say, hey, you can test for asymptomatic individuals yet. I hope that's where we go. I know there's people out there cringing because they know that the sensitivity of these tests are much lower, but we'll talk briefly on how there's different views of that. Because, and as I alluded here, there's these tests range in sensitivity from 84 to 96% in symptomatic individuals. So yes, it is less sensitive compared to PCR. But are there better things to come? As we've seen with HIV antigen testing, we use antibody antigen testing already. So these are the 4 that we're able to glean from the internet. There's probably more upcoming. The best one out there has about 96% sensitivity. Some have been coupled to apps so that you can test and then have that data on an app to show how long ago you were negative. And they're cheap to produce. That's the beauty of this. So I'm not, going to have to fight a tooth and nail to find my extraction kit, my pipette tips, and PCR reagents. But on the flip side, the federal government does buy these up. So we were excited to hear recently that there was a new test that was out there, but we also were sad to know that 3 months supply was already bought up by the federal government for distribution. And this is where I bring the little concept, and it's still a hot topic, still controversial. I used to live in the ivory tower of lab medicine, saying that no test is better than an inaccurate test, but now revise that. How inaccurate is inaccurate are we willing to tolerate during this pandemic and other cases? The notion here from rapidtest.org and is pushed out there by Michael Minna, and feel free to read the articles about that he's published. But the concept here is by the time we test at our institutions with PCR, we're already on the latter end of the viral transmission curve, as you can see here on the right. If we try to test everyone with expensive PCR tests, it's not sustainable. We don't have enough kits, and it's extremely expensive. So the question comes up is if a test that's a little bit less sensitive, say 70% sensitive, and that's not great in the pre-COVID world, but 7 out of 10 you're going to catch. And so in this case, you're gonna be able to test quickly for something that's cheap, widely available, and if you test enough and test frequently, you're gonna be able to catch folks that are in the pre-transmission phase, the asymptomatic phase, and so that helps slow down the actual progression of the pandemic, and obviously complements PCR testing. All right, so we've talked about the front end, we've talked about the back end. Now we're gonna kind of round out the rest of our talk with the middle, which is where staging, prognostication, and therapeutic monitoring plays a big role. So going back to our first patient at UC Davis, obviously they were critically ill, being considered for ECLS. And you can see these are their admission labs. And I'll highlight the ones that were pretty much abnormal, which was all of them. And this goes back to Dr. Lippy's table right here that shows that a lot of these routine chemistry and hematology and coagulation tests we do are great to be able to use for monitoring and see how where this patient's going. And if these are abnormal, it's not really a favorable thing. Even from the complete blood count, more specifically blood smears, our colleague Dr. J.P. Graff at UC Davis, he's a hematopathologist. The first patient in this case, he even knows morphological changes such as leukoerythroblastic reactions that he published. So there's a lot of things going on with these patients. But let's first focus on blood gases. Well, as you can imagine, if you're going to need ECLS because of ARDS, there's going to be acid-base disorders, especially on the respiratory side, and we'll talk about the kidney side very shortly too. And so in this case, most serious cases do require ECMO and at least mechanical ventilation, and so and at the same time you'll see AKI, myocardial dysfunction, and all these, of course, will lead to your blood gas acid-base abnormalities that you're going to have to address. You can see on the right how this plays out with SARS-CoV-2 testing. And as we learn with the Ebola world, that point of care does play a role because it's fast. It minimizes sending samples all over the hospital to send it to a satellite lab, a blood gas lab. You could do this at the bedside to make more real-time management feasible. It minimizes potential contamination. It keeps the samples in that unit, for example. And there's lots of options out there as they evolve. At our institution, we actually switched over to a different blood gas platform mid-pandemic because of supply chains too. So it's actually good to be able to adapt to this. And there's now new systems that are coming out there that are adapting various algorithms that make managing COVID patients a little bit easier. One platform actually has the ability to generate an ABG, arterial blood gas-like result from a venous blood gas. This is specifically for COVID testing. So make collection of these samples easier. No one really likes to do an arterial stick or have A lines inserted, but this helps with the process during this pandemic. Creatinine, of course, we're in the kidney world now, and as I mentioned, COVID-2 does impact the kidneys. And so you see here that the sepsis from COVID-2 in the cytokine storm does contribute to the acute kidney injury, and perhaps also direct viral damage of the kidney itself. And you can see this nice figure here published in the Journal of the American Society of Nephrology. You'll see that the actual virus has many mechanisms to wreak havoc on our kidney, leading to acute tubular necrosis, interstitial inflammation, and the list goes on. And of course, cardiorenal syndrome, which will play back and forth between the cardiac side and the kidney side. So this leads to us relying on our mechanisms to identify AKI early, and this unfortunately relies on serum creatinine and urine output based on the KDIGO criteria. These parameters are good from the standpoint it looks at functional loss, but we do need to try to detect these early, and unfortunately this area of biomarker has not reached the point yet. I think it will get there soon, but it's nice to have biomarkers that are earlier in the process to detect the damage itself. And so these are things that we want to focus on, and specifically this is a paper from Burns that we published last year, and we have another paper that's accepted and waiting to be published on this topic. But in this case, you see that urine output and creatinine have terrible receiver-operator characteristic curves, and the area underneath of which are 0.56 and 0.64 respectively. So really not that helpful. But what we found, and these are in burn patients, just want to clarify, not in COVID patients, but the AKI can be translated over that. We see that a biomarker called NGAL working quite well, and BNP right there, B-type natriuretic peptide, also plays a good role. But I want to focus on NGAL, which is neutrophil gelatinase associated lipocalin, and we were lucky to be one of the early users of this biomarker from a research perspective. You can see our papers going back, I think, back to 2014, if I recall. And NGAL is quite interesting, obviously produced by neutrophils, filters by the kidney. During AKI, plasma levels will rise, and oddly enough, renal tubular cells start making NGAL, which is also then detected in high levels. So this is a biomarker of kidney injury. So this is very nice. And in fact, our burn study in related to burn shock, we were able to predict AKI 4 hours after admission rather than 12 to 24 hours based on just creatinine and urine output. So it does facilitate early recognition of AKI, and there's some discussion out there of using machine learning even to be able to enhance that performance. So you can see here, going back to that panel, that the actual ROC curve has an area in the curve of 0.92 compared to 0.56 for urine output. So quite promising there as NGAL becomes available on the market. Of course, our friend procalcitonin, for those that are members of the AACC and go to the AACC, you hear procalcitonin annually. And procalcitonin, we're one of the early adopters too. And procalcitonin, for those that aren't familiar, it's a prohormone to calcitonin. Produced by C cells. Typically, you hear about it as a bacterial biomarker, bacterial infection biomarker. So PCT levels go high in bacterial bloodstream infections. It seems to work well with respiratory tract infections. For viral infections in particular, PCT levels should be suppressed because interferon gamma that's released actually suppresses the production. It should be low in non-specific inflammation, neutropenia, and viral and fungal infections. You can see here this graph, this plot that we put together comparing PCT over time for sepsis with other biomarkers. It rises quickly in about 2 to 4 hours, half-life about 24 hours, peaks in about 8 to 24 hours. And if you are treated, had adequate treatment for your infection, it should decrease at least by 50% and return to normal upon recovery. And as you would expect in COVID-19 patients, the majority of patients have very low PCT levels, especially in the early phases, right? So it's because it's a virus, it's not a bacteria. But interestingly enough, up to 35% of cases in literature have suggested that PCT levels are abnormal. And this includes our first patient at UC Davis. If you remember our previous slide, PCT was abnormal. It's been hypothesized that this could be due to tissue injury and or bacterial co-infection, tissue injury being ARDS, AKI, list goes on, but perhaps bacterial translocation and other factors are playing a role that are prompting a rise in PCT. But at the end of the day, an increased PCT is associated with a nearly 5-fold risk for COVID-19 severity. So very interesting use of that biomarker there that continues to evolve. We also see C-reactive protein playing a role, so an old biomarker, but in this case, this is from the actual damage from the various tissue and activation of complement. but we see high CRP observed in 86% of patients with severe COVID-19. So this is another biomarker that we see. And you can see here with CRP that it is significantly higher in non-surviving COVID-19 patients versus survivors there. Another set of biomarkers that we want to look at is IL-6, and you can see here interleukin-6 plays a role in cytokine release syndrome. I know our California Department of Public Health guidelines include IL-6 as a biomarker to help work up individuals, and specifically children, with the multi-inflammatory systemic response in children syndrome. And you can see here that it could provide value. And there's some discussion, and unfortunately I'll have to update you all on bad news that these drugs unfortunately don't play a role, but there was a lot of early discussion of using IL-6 blockade to be able to help with mitigating the adverse effects of COVID-19, but monitoring perhaps may be useful too, to help risk stratify these individuals out there. So so you can see here the premise comes from studies that show that that non-surviving COVID-19 patients had really high levels of IL-6, and of course versus survivors there. This led to the discussion: should we be using these biologic drugs to block IL-6 binding? And the data here showed some promise, but unfortunately, phase 3 trials for this drug, as well as I believe this other drug, did not show promise. So I don't think that has made the same traction as we saw with other drugs that are now making their way through the actual exploratory as well as FDA EUA pathway. There's now continued evolution of our understanding of the virus, and you can see here that there's discussion that the bradykinin pathway can actually play a role as well. This is from a study from Oak Ridge National Lab that showed that it appears that the virus could upregulate ACE2 receptor expression in other organs, and that may explain why this virus is affecting so many different parts of our body, including the bradykinin pathway, which then activates the coagulation cascade, which in this case shows or partially explains why D-dimer levels are also higher in both normal patients and surviving patients versus non-surviving patients, just higher in non-surviving patients. There's some studies out there that say that even surviving patients who are not too symptomatic have elevated abnormal D-dimer levels. And you can see here this figure right here showing how CoV-2 various complex pathways with coagulation that links to coagulation, including the kallikrein-kinin system, to ultimately produce that D-dimer. But yeah, you can see that the coagulation cascade plays a big role, which of course leads to all that systemic problems we see. So it comes back to all these two right here. So to close out, the COVID-19 pandemic has created unique challenges globally, with laboratory medicine playing a very central role. Diagnostic testing consists of molecular and antigen testing at this time. There's a lot of innovative studies going on. Perhaps other technologies may come out. But antigen testing is slowly emerging as a potential low-cost adjunct. I want to emphasize adjunct to molecular solutions. You're not going to replace molecular at this time. The value of COVID-19 serology testing remains limited, but more investigations are limited. And of course, as vaccines become available, we will see it come back with better uses and more refined approaches and a better understanding of immunity. Biochemical biomarkers play an instrumental role for staging and prognostication and for therapeutic drug monitoring. Abnormal procalcitonin, CRP, IL-6, coagulation, and renal parameters are often encountered in COVID-19 patients that are having severe responses. And then COVID-19 related respiratory and metabolic reagents require blood gas testing, and point of care blood gas testing provides additional value because, it's point of care, it's fast, and can be contained within the patient's room. So I will stop there, and I'm now happy to take questions.
Thank you, Dr. Tran. We're now at the Q&A portion of the session, and as everyone gets their questions together, I can start us off with a couple of questions. So first off, what do you think the applications are of some of these tests in the outpatient setting, for example, for prognostication in terms of, there's a lot of debate about D-dimers and CRP, PCT. What are your thoughts on future applications and the direction for predicting who might need to be hospitalized?
Yeah, that's a great question. I would say that I'll speak to it from the standpoint that I do see potential value, but at the same time, it's how the patients present at these outpatient clinics, right? If they're well enough to walk into your door or through your drive-through, drive-through testing, that still brings up a lot of questions. And I would probably say that the other perspective as a laboratory, most places don't have the ability to do D-dimer and a lot of these other tests at the point of care, at the clinic, right? Our health system has 17 clinics out there, and, it takes a while for samples to get back to the core lab to get results back. So it's all about what you're going to do with that result and how fast those results are there to take action on. I think where things may go are these antigen as well as rapid point of care molecular tests for COVID-2. So, for example, you'll be able to, in part, as inhaled remdesivir becomes available, and if it shows promise, you could perhaps see high-risk patients who are not necessarily symptomatic, but at the same time they're PCR positive. They can be placed on these drugs more quickly. So I think, again, it comes back to, what are you going to do with the result and how fast do you get to take appropriate action. That's what matters, especially with a lot of places now trying to minimize in-person visits and, of course, drive-through testing. A lot of these bread and butter D-dimer, procalcitonin, CRP may not be readily available, or at least readily be available in a timely fashion.
Yeah, we certainly struggle with that even in the emergency department, whether any of these are of utility, particularly if a patient's going to be discharged. My second question is, what would you say was the greatest challenge of implementation of testing to support SARS-CoV diagnosis or management of these patients over the last couple months?
I would probably say that it's obviously reagents will be my first, reagents and the ancillary supplies for our PCR test, right? And, the AACC has definitely done some surveys to even, to quantify that. And a lot of hospitals out there are always short. We never, not many places can really achieve their full true maximum capacity due to these reagent shortages. And I think that's been our challenge to be able to scale up, because we always want to test more. And the second issue, like with a lot of labs out there, and maybe more, our modest lab, our institution, it's staffing, getting more staff, and we've been so lucky at our institution to get more staff brought in. But bringing additional staff to expand testing over these many months, and the personnel have been heroes to be able to get testing up so quickly. But it does take a toll on our laboratorians. We've been in a continuous validation state since February 29th, and we are continuing to validate new things. And I think the challenge now is that we went into this with a sprint and realizing it was a marathon. And it's kudos to all the amazing laboratory personnel that continue to fight and continue to do amazing things. But that's been my concern and continuing concern, that how can we continue to sustain operations as supplies wax and wane, and as potential increases in testing demands grow as we head into flu season.
Okay, thank you. So I'm going to try to get to some of the questions that are on the Q&A session here. So, we all know that there are different phenotypes of patients with COVID-19 that are sick enough to be admitted to the ICU. And, what's been talked about is the phenotype of the happy hypoxemic versus the classic ARDS picture. So the first question is, what are the laboratory characteristics of cytokine storm associated with severe COVID-19 that's sick, for where patients are sick enough to be in the ICU? And then I'll add to that, can you talk a little bit about the bradykinin hypothesis?
Sure, and I'll answer the first question first. Is that this will be a lot of presentations very similar to obviously sepsis, the ARDS, multi-organ dysfunction that occurs from that, all the cytokines that are being thrown out there at you, right? So, and that's one of the challenges with this process, is that it's hard to identify, especially since most of our places don't even have multi-cytokine testing. It's a send-out test at best. And that's why IL-6 has become very important. I know that there's at least one. I know there's at least a couple more IL-6 assays that are showing up. The volume at our institution may not be high, because there's still just few of these patients that show up presently, and I hope they remain that low. But I think that's where these tests play a big role. So they'll present similarly to other cytokine storms. I mean, sepsis being a classic one, especially severe sepsis, septic shock, but also similarly, and that's why a lot of drug companies went down this rabbit hole of cytokine storms following CAR T-cell therapy. So I think those are the presentations that you would typically see. For the bradykinin pathway, I think what I presented, and I will say I'm not a coagulation expert, but at the end of the day, it just seems that this bradykinin, as we all know, connects with so many parts of the coagulation pathways. in this case, once the ACE2 receptor upregulates that, you'll just see this increased degradation of the fibrin and the FDPs, fibrin degradation products, and of course D-dimer at the end. That serves as a biomarker of all that coagulopathy and coagulation issues that are ongoing due to the virus.
So that leads into the next question is, at what stage would you consider starting anticoagulation therapy in COVID-19? I guess I can address that briefly myself. So I think this is still controversial because there's a lot of evidence coming about that COVID-19 really affects the vascular system. It's not simply a respiratory disease. And even though there are ongoing trials right now and the evidence is evolving, I believe that most hospitals are recommending, where it's not contraindicated, to start prophylaxis for VTE in patients that are sick enough to be hospitalized with COVID-19. So that's just a quick response. So more to come. So
it's
here,
ever evolving, yeah, unfortunately.
So there's another question regarding whether clinicians are using a scoring system with these laboratory parameters to judge the severity of COVID-19, and is that something that's happening yet, or is there research ongoing there?
I believe there's been a couple scoring systems. I think one in the UK that people have looked at, but I don't think it's widely adopted yet. I know there's people working on, and one of my colleagues is looking into this with a big data, look at machine learning. So I'm not too much of a fan of a scoring system. It's nice, quick, and dirty. Don't get me wrong. There are uses for it. We obviously use it for other things in practice. But I think this is such a complex disease that a scoring system may not yield as much value as we would want versus running a PCR test to diagnose someone and looking at other parameters to see if they're getting worse or not.
Okay, so I think we have time for a few more questions. So this is a great question. So as we're approaching the influenza season, what are the changing recommendations or guidelines for including rapid respiratory testing for other viruses than COVID-19?
Well, I think from my end, maybe you also have feedback on this because I know you and I have talked about this greatly, but I think for the most part, flu and COVID-2 testing are probably extremely important, and even our feedback from our clinicians across the health system, they would love to have the combined flu plus SARS-CoV-2 assay, and luckily we are shifting towards that, and so we are looking at bringing up a flu plus SARS-CoV-2 high-throughput assay on our high-throughput platform, and we are adopting a point-of-care flu AB SARS-CoV-2 assay for our EDN clinics too. So we're going to try to be as well stocked as possible. It's not as it's not unlimited, but I think that's where it's at. Now the question of the rest of the respiratory panels, there are two challenges with that right now. Is one, what's the value of the 18 other viruses that our assays can detect that aren't flu or COVID-2, with the exceptions of RSV and some others that are for special populations. But also the price tag for some of these big panels too. So it's not necessarily for inpatient issues, but for outpatients. And some of these people with different insurance plans, they're going to get hit with a big bill. And what are you going to do with that result, right? And those are the questions there. But for the most part, for the rest of the panels, I think my recommendation would be if a patient's at risk and being admitted, that's fine to do the full panel if that has value. But for the most part, it seems that our bread and butter for fall and winter will be flu and COVID-2.
Yes, I think I can add to that. Considering the outpatient setting or the emergency department, I think during this influenza season, and it remains to be seen how severe it's going to be depending if we follow the pattern of the southern hemisphere, but I don't see a situation where we won't want to test for influenza also. Namely, it is possible that we would have a patient that is negative for SARS-CoV-2 and positive for influenza that would provide an alternate diagnosis. We do know some of these patients have co-infections, but I think it's less likely if you have negative for one and positive for the other. And it also may impact management for patients that are sick enough to be admitted to the hospital. I agree with you that the other viruses, while interesting from an epidemiologic perspective, given that most of them, supportive care is the only marker, I don't think there's much utility for outpatients, at least, perhaps as part of a workup of a patient who doesn't have an alternate diagnosis. But I think the cost in the outpatient setting, we've seen time and time again in some of the own research we've done doesn't really seem to help. So I think we only have time for one or two more questions. So I just want to, let me just scroll through here. So there are concerns, obviously, with diagnostic testing, as you mentioned, with reagents. So one of the questions is, related more to screening asymptomatic patients pre-procedure or pre-hospitalization. What are your thoughts on that? I do know we're doing that for all of UC Davis, but what are your thoughts? Should we get in a situation where we don't have sufficient testing?
I think, that's a great question. And I think that's why pooling is so interesting to us in other places too, is because if you're doing asymptomatic screening especially for pre-procedure related surveillance, pooling does work because and I'm a proponent of pooling, or we're working on pooling. And so in that case, I think that plays a role where, for example, our platform, if we do a pooling density of 5, we're able to do about 470 tests per batch, and that assumes a prevalence of less than 5% to maintain efficiency. And our prevalence right now, my understanding, is about 1% or less in that surveillance group. So it works quite well. In fact, you can pool even more than that, assuming the FDA allows you to use that higher pooling density. So I think there's still a value for that because it does 3 things. One, our providers are more comfortable. It does help reduce PPE in some cases, and those are all things that we have to think about. However, with that said, to pool, you run a little bit slower. You have to accrue sufficient samples to pool, especially on the high throughput batch systems. So there is a turnaround time delay. I know a lot of clinicians out there want to test the day of, which is not going to be feasible. And some folks want to test the day before, but sometimes it takes half a day to batch that much, depending on how many samples you get in. So those are the trade-offs that you have to think about. But I am supportive of the asymptomatic testing. I am supportive of the pre-procedure testing. But we shouldn't be testing necessarily everyone until we do have an unlimited supply of reagents. So we do have to be mindful and be stewards of this limited and valuable resource we have.
Yes, and certainly I think it's going to depend on the prevalence, in our area and what that predictive value is going to be of that negative or positive test. So unfortunately, we're running out of time. I think I'd just like to take one last question, which is also something of great interest on my end also, is could you please comment on point-of-care antigen testing and its feasibility and what the state of that is right now?
Yeah, it's very interesting. I'm quite fascinated by it too, and we ourselves are doing our own studies. I'm sure other places are too, looking at various kinds of antigen tests that are out there that we can get our hands on. I think that's going to have the ability to, like I said, augment our existing PCR capabilities. And the question comes up is, how sensitive are they going to get? And data looks promising as they get more sensitive, but again, among symptomatic individuals. I hope that at some point that there's data out there that support the use in asymptomatic screening. So this is where these cheaper tests, assuming they perform well, I want to emphasize that, they are very useful for that return to work, return to school capability, because rather than investing millions of dollars on a PCR system to screen everyone, you now have these test cards or whatever that you can use very quickly. And I know the FDA a little bit over a month ago released guidance on over-the-counter and home testing claims. So that paves the way, assuming any manufacturer chooses to go down that pathway, but it paves the way that perhaps these tests could be used for that. Obviously, a lot of us in the laboratory will cringe a little bit. How do people document? What are people going to do with those results? I agree. Those are things we have to overcome. But the challenges right now is, we just don't have enough testing, and testing is not necessarily always being utilized where it should be. So we need to have more options and have more flexible capabilities.
Yeah, I agree with you. I think this is more of a commentary than anything, but, I have seen some studies suggesting that even with the lower sensitivities, when applied in a community, that it could significantly decrease transmission from person to person. I think it's unfortunate, if I may just say so, that all of this was deployed in the settings, that make the least sense, like, for example, nursing homes and long-term care facilities, where, you really are going to want to confirm those negatives with a PCR, and also the struggles of these sites to really, once they have CLIA, to actually be able to execute this point-of-care program in the nursing homes. In public health, we're struggling a bit with that right now. How are these going to be reported, the need to follow up for negatives, the sensitivity not being high enough, for high-risk healthcare workers that are asymptomatic. So I think it'll be really interesting to see how this evolves, and it would be great if, some of this could be accessed by lower risk populations, such as young adults or schools where we would be testing symptomatic people, and it might actually, have a bigger public health impact, outside of those long-term care facilities. So I think, unfortunately, we're out of time. I know there are lots of great questions, a couple that we didn't get to, but I want to conclude now this portion of the session. So I think, we'll turn it over to the AACC team.
Thank you, Drs. May and Tran. This now concludes today's webinar. Thank you all for participating. And again, we want to thank Radiometer Medical APS for making this educational webinar possible. We hope you all have a wonderful rest of your day.